Scientists have long been fascinated by how animals such as zebras, leopards, and snakes develop their striking skin patterns. For decades, the dominant explanation came from the British mathematician Alan Turing, who proposed in the 1950s that developing tissues produce chemical agents that spread out, react, and inhibit each other, forming the basis of animal patterns. These are now called Turing patterns. However, when scientists simulated this model, they noticed that patterns produced were blurry and lacked the sharp boundaries we observe in nature. This left a major scientific gap. How do real animals achieve such precise, crisp pattern? A breakthrough study from the University of Colorado Boulder, published recently in Matter, provides a compelling answer. Researchers Ankur Gupta and Syamak Mirfendesky added a new physical phenomenon called diffusiophoresis, discovered by Gupta's team in 2023. Diffusiophoresis describes how tiny particles suspended in a fluid attract or repel each other like magnets, causing them to clump together. When they applied this mechanism to the classical Turing model, the patterns became significantly sharper. However, there was still a mismatch. Nature is imperfect, while the simulations looked too symmetrical. The researchers refined their model further by assigning realistic, varied cell sizes and observing how these cells moved within tissues. This proved to be the missing piece. Cell size directly influenced how the patterns formed. When cells were very small compared to the pattern thickness, they packed neatly, forming smooth and symmetrical designs. But as cell size increased, cells began bumping into each other. They no longer fit perfectly, creating irregularities, distortions, and broken boundaries, exactly the kind of imperfections seen in real animal coats. Larger cells also produced broader and more irregular patches, closely resembling natural biological variation. This means that tiny inconsistencies in animal patterns can be explained simply through differences in how cells move and clump. The model still has limitations. It assumes cells are hard spheres and does not include biological forces, such as adhesion. Yet, it marks a major step toward understanding natural pattern formation and has potential applications in camouflage design, textiles, and developmental biology.